Cellular Respiration by Tristan, Sarah, Dayanara, and Gianna.
Cellular respiration is the transfer of stored energy within cells into molecules of adenosine triphosphate or ATP. Cell respiration can occur either with or without the presence of oxygen, known as aerobic or anaerobic.
Glycolysis discussion points. Glucose, oxygen, carbon dioxide as it is a waste product, pyruvate, NAD+, NADH, ADP/ATP, substrate level phosphorylation will all be discussed.
Either way, cellular respiration begins with the anaerobic process of glycolysis, which produces 2 3-carbon molecules of pyruvate for every molecule of glucose it breaks down. Glycolysis occurs in the cell and produces 4 molecules of ATP, but because its energy of activation is 2 molecules of ATP, resulting in a net gain of 2 ATP molecules. These ATP molecules are produced by what is called substrate level phosphorylation. It is important to know that this occurs in the cytoplasm. Substrate level phosphorylation is a direct enzymatic transfer of a phosphate to adenosine diphosphate, or ADP. The amount of ATP produced, some substrate level phosphorylation and glycolysis, however, is relatively small. Much more ATP is produced in a later step of cellular respiration, before entering the Krebs cycle the pyruvate generated in.
Link reaction and Krebs cycle discussion points. Pyruvate, acetyl-CoA, FAD, FADH2, mitochondrial matrix will all be discussed.
The link reaction.
In the mitochondrial matrix, pyruvate and coenzyme A combine to make acetyl-CoA, and per pyruvate, the byproduct of NADH is produced. The acetyl-CoA produced enters the Krebs cycle.
The Krebs cycle.
This is an enzyme-catalyzed rotating cycle that breaks down a carbon molecule to produce molecules that are used in the electron transport chain. The cycle releases 3 molecules of NADH, 1 molecule of FADH, 1 molecule of ATP, and as a waste product, carbon dioxide. The cycle begins with acetyl-CoA combining to form citrate. Citrate is oxidized and decarboxylated to form acetylglutarate. Acetylglutarate is further oxidized and decarboxylated to form succinate. Succinate is oxidizes and regenerates as oxaloacetate.
Oxidative phosphorylation. The electron transport chain, electrons, NADH, NAD+, FADH2, water, chemiosmosis, proton gradients, seriste, inner and outer mitochondrial membranes, proton pumps will all be discussed.
Oxidative phosphorylation is a process that is primarily for producing ATP for the body's energy. Its first part is the electron transport chain. Its main purpose is to form an electrochemical gradient. This system is made up of protein complexes. NADH approaches the first complex and donates its electrons, represented by the red models, which results in NAD+. When the electrons enter complex 1 gains enough energy to pump protons, or H+, represented by the blue models, from the mitochondrial matrix to the inner membrane space. The electrons continue to travel through the transport chain. FADH2 approaches the second complex and donates its electrons to it, and in the process becomes FAD. The electrons move into complex 3, and their arrival allows complex 3 to pump more protons into the intermembrane space. The electrons then travel to complex 4, where, again, protons are pumped into the intermembrane space. The electrons are ultimately accepted by oxygen, which turns into H2O molecules. In chemiosmosis, ADP is transformed into ATP. At this point, there is a proton gradient formed by the electron transport chain. The protons follow the gradient and go from the high concentration area through the ATP synthase and back into the mitochondrial matrix, where they are used as an energy input for the reaction of ADP into ATP. Some important things to know are that the cristae are the folds of the mitochondrial membrane that increase the capacity to produce ATP energy. It also increases the surface area for oxidative phosphorylation. The inner and outer mitochondrial membranes act as transportation systems, where protons move across the membrane to create a proton gradient. The outer mitochondrial membrane essentially acts as a barrier to maintain the proton gradient established by the inner membrane. All of this is essential for ATP synthesis.
Anaerobic respiration discussion points. Note that oxidative phosphorylation, electron transport, the Krebs cycle are all components of aerobic respiration. Now, the components of anaerobic respiration include alcoholic fermentation, lactic acid fermentation, glycolysis in terms of its products, lactate, electrons, and replenishment.
Anaerobic respiration. When defined, it is the biological process where cells produce energy ATP without the use of oxygen. There's two types of this respiration: lactic acid fermentation and alcohol fermentation. In alcohol fermentation, microorganisms convert sugars into alcohol by producing energy in the form of ATP. After glycolysis and the production of pyruvate, this molecule is decarbonized, forming acetaldehyde. Acetaldehyde is then reduced by the enzyme alcohol dehydrogenase using electrons from NADH to make ethanol and regenerate NAD+. In terms of lactic acid fermentation, it occurs as follows. The glucose in glycolysis is converted to pyruvate, and it releases a net gain of 2 ATP molecules. The NAD+ is converted into NADH. Then the pyruvate is reduced to lactate with the help of the NADH, which is produced in glycolysis. NADH donates electrons to pyruvate, converting it into lactate. This reaction also regenerates NAD+, making it replenishable. Here's a diagram that helps summarize every major topic discussed in this video. That is the end. Thank you for your time.